Now we will see genetic code. Okay, genetic code. So we have discussed everything about replication of DNA. That is, during the cell division, when the cell divides into two daughter cells, along with it, the equal amount of DNA also divides. So the DNA, they pass equal amount of DNA to two cells. Okay. So from that, we need to discuss the transcription and then translation. We have also seen semi-conservative also. Okay. So from the DNA strand, now protein will form. So for the formation of protein, the cell has to undergo transcription process. Okay. That means from DNA strand, mRNA will form. That means DNA will give information and the mRNA will carry that information. That is the messenger RNA. We have discussed when we have discussed the mRNA in short, but again in details, ekdam details we will study in transcription process. How it actually happened, how it actually takes place, how from DNA strand the mRNA is formed in actual process. The details we will study. Then from transcription, once the mRNA carries all the information, all the required information means what information they carry? They carry all the nitrogenous base pair, nitrogenous bases from DNA. Okay? Adenine, guanine, there will be no thymine because that is the mRNA, uracil, cytosine. They will carry all those nitrogenous bases in the form of triplet. We call that it as a genetic code. Okay, we will study. We call it as a genetic code. For the formation of protein, what things are required? Amino acids are required. Genetic code are required. Genetic code means nitrogenous bases. All these things are required. So first of all, the mRNA has to carry information from DNA strand. Information means nitrogenous bases. That is the genetic code. Okay. So once the mRNA is formed, the process is called transcription. Now that mRNA will undergo into the translation process. The actual formation of protein will take place here. How? We will see. But before this thing, we need to see the genetic code. Genetic code. Okay. We can also discuss first transcription, then genetic code. But let me first introduce genetic code before going to this process. Now this process is known as central dogma. Okay, this complete process is called it as a central dogma. I could have discussed genetic code before discussing transcription process, but you will get confused. So that's why I will first go with the genetic code, then we will discuss. It feels so good talking to myself alone. It's better. It's like that only. So genetic code, we need to see some important features along with what are the genetic codes, which codes are they going to carry, who is going to carry mRNA, so that we will see. So first of all, important features. Okay. Now genetic code was first discovered by a scientist, George Gamow. Yeah, he was the first scientist who discovered genetic code. All are males. Okay, all scientist names are male. There is no female. So genetic code was first discovered by George Gamow. Now we're going to first write some important features of genetic code. Okay. Next, there are 4 nitrogenous bases. Actually, there are 4 nitrogenous bases which makes a genetic code. Okay. 4 nitrogenous bases are responsible for genetic code. You write complete sentence in your own language. Okay. There are 4 nitrogenous bases which makes a genetic code. They are namely, bis means what? Namely, in short form. BIZ. Adenine will be there. There will be thymine. No. Thymine get replaced with uracil. Because genetic codes are the codes that is going to carry by mRNA from DNA. So adenine will be there, but it is a mRNA, RNA. So the thymine will get replaced by uracil. Okay. So cytosine and guanine will be there. So actually 4 nitrogenous bases are there which makes a genetic code. These 4 nitrogenous bases makes total 64 genetic code. How many? 64. We will see that. So 4 nitrogenous bases are the actual genetic code. Means they are going to make the genetic code. Okay. Now total genetic codes are 64 genetic codes. So how many total genetic codes are there? So total 64 genetic codes. 64 genetic codes, huh? There. Alright. So 64 genetic codes are there. How come 64 from 4 nitrogenous bases? We're going to see that. Don't worry. So out of 64, 61 codes for 61 genetic codes for amino acid. Okay. सब genetic codes amino acid के साथ bind नहीं होता. Say for the formation of protein, the amino acid are required as well as genetic codes. You know what they do? The amino acids, they bind with those genetic codes. We're going to see that when we discuss translation. But here, just remember the features of genetic code. Okay. So out of 64 genetic codes, how many types should I say this? 61 codes for amino acid. 61 codes for amino acid. What happened to the rest 3? The remaining 3 codes, that means genetic codes, are called as stop codon or nonsense codon. Nonsense codon, it means it doesn't make any sense. Okay. Out of 64, 61 codes for amino acid and 3 codes for stop codon. It's going to stop the process of translation. We will see that. Or we also call it as a nonsense codon. That means these 3 genetic codes, it doesn't code with any amino acid. That's why it doesn't make any sense. That's why nonsense codon. But it has a role to play. Okay. We will see that. Buji paya sa? Okay. Next important features. See, approximately 20 amino acid is required for the formation of protein. Okay, 20 amino acid is going to bind with 61 codon of genetic code. Yeah. but the 3 genetic code will not going to bind with any amino acid. Okay, we will see that. So let me just stick with this. All genetic codes are non-ambiguous. What does that means? That means all genetic codes are specific. Specific means they're going to codes for only that particular amino acid which is going to form the protein. Okay, that's why we call it as a non-ambiguous. That means all genetic codes are specific. Remember all these features, that is very important. Before going to the process of transcription and translation, you must know the roles of genetic codes. Now, next, when I talk about codes, that means genetic codes only. Let me write here, all genetic codes are comma less. Whenever we write genetic codes, there is no comma, or we are not allowed to write any comma or full stop, apostrophe s, anything. All genetic codes are comma-less. Oh, one more important. All genetic codes are read in triplet form. Okay, so we have a 4 nitrogenous base pair. 4 nitrogenous bases which actually makes a genetic codes. That means 64 genetic codes. Now when we read all those genetic codes, we read in a triplet form. Triplet form, suppose we have okay, so this we have nitrogenous bases. Suppose, so whenever we're going to read it, we're going to read it in a triplet form. AUC, GAU, CGA, UC, there is one more, suppose UCG. So like that, we're going to read in a triplet form, not in a doublet, not in a singlet. Okay, we're going to read in a triplet, okay, three words, triplet form. We have taken the example of when we study about the friendship mutation, right? Why did the cat eat the rat? Why did the cat eat the rat? Okay, so we have taken that example during friendship mutation. Okay, so whenever we read the genetic code, we read in a triplet form. And suppose one genetic code is missing here, so it will result into mutation, some kind of disorder or diseases. So whenever we read genetic codes, we read in a triplet form. All right. Okay, coming to next. All genetic codes are non-overlapping. Okay, all genetic codes are non-overlapping, not with one another like that. They are in a serial form. Okay, serial form. All genetic codes are, one more thing, universal. Okay, universal. All right, so these are the important features you need to remember genetic code. So how many genetic codes are there? 64. But normally we have the 4 nitrogenous bases makes the genetic codes. 64 genetic codes are there, which are 4 nitrogenous base pair AUCG. These are the 4 nitrogenous bases which are going to make the genetic code. How many genetic codes? 64 genetic codes. Out of 64 genetic code, 61 is going to bind with the amino acid for the protein synthesis, but the remaining 3 genetic code, 61, 62, 63, 3 genetic codes will not going to bind with any amino acid. That's why we call it as a stop codon. Why stop codon? We will see. And also we call it as a nonsense codon. Nonsense. Okay, these are the some features. Well, let me give you example of stop codon here. It is important, okay? UAA UAG, UGA. So these are the three stop codon or nonsense codon. Remember UAA, UAG, UGA. Okay, whenever the amino acid, suppose this is amino acid, they're going to bind with, suppose this is your genetic code. They're going to bind it, and during the process of protein formation, that is during the translation process, they're going to slide over the ribosome. When they slide over the ribosome, they're going to bind all the genetic code, others genetic code, that means, okay, they're going to they're going to continue their process until and unless the stop codon comes. Whenever these three things, any of these three things comes, they're going to stop the process. They're going to force stop. That's it. Okay. Now one more thing. Start codon. Let me write a start codon. Example of start codon. Start codon means the first codon which is going to come and bind with the amino acid. The first is codon. That is start codon. The example is AUG. The first codon. What? AUG. Okay. That is the example of first start codon. So this question came this year, but unable to answer it. I don't know. They are unable to answer it. Some of them. So AUG. Yeah. So now we will see how come 64 codon. So normally it is controlled by 4 nitrogenous bases. AUGC. A means apple. Right? A means what? Adenine, U, uracil, cytosine and guanine. Why uracil? Because when genetic codes are made, it is made from the DNA strand by mRNA. So mRNA is going to make the genetic code. They are going to code with the DNA strand. They are going to carry the information. So that thymine get replaced with uracil. A 64 codes. 3 codes are nonsense. So we're going to make a table. So we have 4 nitrogenous bases. So 4 into 4, it is going to have 16 table. 1, 2, 3, 4. There is a process to find out all the 64 codon. There's one more. Okay. First, you write here A, U, C, G. This is first step. In second step, similarly, you put A, U, C, G. Okay. In third steps, you make A, U, C, G. Okay. Now, when we make a cross, this A, first you make a cross with this A, and this A. Okay, this A, and this A. Done. Then you make next A, U. Then let me try. See, A. It's too long. A. Okay. Then again A, U. Okay. Then again A, C. Again A. You will understand. So this work is done in this box. Coming to this U. A, U, C, U, G. Okay. So like that. I hope you are getting this. I hope you are getting my point. Okay. So for A. So this is our stop codon. So like that, you will get 64 code, 64 codon. Now out of 64, you will find stop codon, 3 stop codon. So example UAA, UAG, UGA. Here we have found 1 stop codon. Okay? Oh, 2 stop codon we have found here. So we will found 3 stop codon like that. Okay? So that's how we're going to do the genetic code. That's it. Okay? In next class, we're going to discuss transcription and translation process. All right. In next video.